Welcome to the Deep Dive. Our mission today, well, it's about digging into an agricultural innovation that sounds almost too good to be true. We're talking about turning everyday agricultural waste into, believe it or not, a high-tech shield for olive trees, specifically using things like pomegranate husks and carob leaves. We're diving deep into a fascinating new study from researchers in Spain and Portugal looking at how these natural extracts can fight off one of the worst diseases hitting olive groves.
And it's crucial to understand just how big a deal this disease, anthracnose, really is. Olive growing isn't just farming, it's ancient, it's culture, especially around the Mediterranean. But anthracnose is a constant threat. It just devastates crops, particularly when it's wet.
Yeah, and it's not just fewer olives, right? It hammers the quality too, which must be terrifying for olive oil producers.
Oh, absolutely. The economic hit is double because the oil quality just tanks. And there's actually a pretty memorable, if unfortunate, name for it. It's often called the soapy olive disease.
Soapy olive, why that?
Because the infected olives get this weird, greasy, almost, well, soapy look. It sounds strange, but it's quite distinct. It makes the fruit unusable, drops the price, and in bad years, wipes out yields. Huge risk.
Okay, let's unpack this then. The enemy here is this fungal disease, anthracnose, caused by several species in the Coletotrichum family. Is that right?
That's the one, Coletotrichum. It's a really widespread and difficult group of fungi, and traditionally, farmers have relied heavily on chemical fungicides to keep it in check.
Which brings us right to the core of why this new research is so timely, doesn't it?
Exactly. Because those traditional chemical options, mainly the copper-based ones, are facing major restrictions. Especially in the EU, there are serious environmental worries about copper building up in the soil.
So farmers are kind of stuck. They need something effective, but the old standby is being phased out.
Precisely. There's huge pressure to find alternatives that work but are also, eco-friendly. And that's where this study comes in.
And the solution they're looking at isn't some complex new chemical. It's literally leftovers, waste from pomegranates and carob trees.
Yes. And what's so neat about this, what's fascinating, is how it fits into this idea of a biocircular economy, taking stuff that the food industry or agriculture would normally just discard.
Like husks and leaves.
Right. Stuff considered waste and turning it into something valuable, a bioprotectant. It's not just finding a green alternative, it's actually closing the loop, using co-products instead of creating more waste. It's smart.
That whole concept, waste becoming a weapon, is really compelling.
Yeah.
But okay, let's get into the science. How does a pomegranate husk actually stop a fungus like Coletotrichum? It sounds like magic, but there must be a mechanism.
There is, and the researchers, Begonia Anton and Carlos Augusti's team, they pinpointed a really clever dual action mechanism. Think of it like attacking the fungus on two fronts.
Okay, two fronts. What's the first?
The first target is dispersal. The extracts significantly knock down the production of the fungus's spores. They're called conidia. And maybe even more importantly, they stop those conidia from germinating properly.
So fewer spores being made,
Yeah.
and the ones that are made can't really start growing, like stopping the seeds from sprouting.
Exactly. If the spores can't germinate effectively, the disease can't spread through the orchard. It cuts down the invasion force, you could say.
Makes sense. Cut down the numbers.
Yeah.
What's the second line of attack then?
The second mechanism is more direct, almost like disabling the fungus's tools for breaking in. The extracts blocked the formation of something called a pressoria.
Opressoria. What are those?
An opressorium is like a specialized pressure pad or drill bit that the fungus develops once a spore lands on the plant surface. It builds up pressure and uses it to punch through the plant's outer layer, the cuticle, to actually start the infection.
Wow, so the extracts stop the fungus from even getting inside the plant tissue.
That's the idea. By blocking the opressoria, you stop that initial breach. The researchers specifically called out conidia and opressoria as the two key structures for dispersal and infection. Hit both, and you really cripple the pathogen.
Okay, this is where it gets really interesting, Franny, because the study didn't just find that the extracts attack the fungus. They also seem to boost the olive tree's own defenses, like a pep talk for the plant.
That's a great way to put it. Yes, especially the carob extract. When they sprayed it on the leaves preventively, a foliar application, it acted like a trigger. It essentially woke up the plant's own natural defense systems.
How does that work? What's happening inside the plant?
Well, it seems linked to the plant's antioxidant response. Think of the antioxidant system as the plant's internal alarm for stress. Could be pathogen attack, could be drought, whatever.
Yeah.
When the carob extract lands on the leaves, it seems to trip that alarm. The plant thinks, okay, something's up, better prepare. And part of that preparation is ramping up the production of certain defensive chemicals,
What kind of chemicals?
specifically certain phenolic compounds. Phenolics are really important molecules for plants. They help with structure, but they're also key players in chemical defense against pests and diseases. So, by boosting these phenolic compounds, the extract is essentially helping the plant strengthen its own armor, making it naturally tougher for the fungus to infect, even if some spores get through.
So it's a double whammy. Weaken the attacker and strengthen the defender. And all from stuff we normally throw away. That's
Yeah.
pretty elegant.
It really is a multi-pronged approach using natural, readily available materials.
Right, but the proof is in the pudding, isn't it? Did it actually work? They ran tests, I assume, in labs, on seedlings.
Oh yes, rigorous tests. In vitro stuff in petri dishes, bioassays on actual olives, and then trials on young olive seedlings in controlled chambers. They needed solid
So what
members.
did they find?
Well, in those controlled seedling trials, the carob leaf extract specifically showed a measurable impact. It reduced the incidence of anthracnose by 35%.
35%. Okay, that sounds significant, but maybe not a silver bullet yet.
Exactly, and the researchers, particularly Carlos Augusti, were very clear about this. 35% is good, it's proof of concept, but it's definitely lower than what you'd get from a copper fungicide, which might give you, 80, 90% protection or more in some cases.
So there's still a gap between this natural option and the traditional chemical one.
There is, but you have to weigh it up, right? 35% protection from a sustainable, non-toxic waste product versus near total protection from something that's facing restrictions due to environmental impact.
That's a fair point. It's a massive step in the right direction, even if it's not perfect yet.
It absolutely is. It shows these bioprotectants have real potential. The next stage is optimization, figuring out how to maybe concentrate the active compounds or combine extracts to boost that percentage closer to the chemical standard.
So this study definitely opens doors. But taking something from a controlled lab environment to, say, a huge olive grove in Greece or Italy, that's a big leap.
It is, and that raises the really important questions about what needs to happen next before anyone starts spraying pomegranate juice everywhere.
Right. What are the hurdles?
Well, the first, and maybe the biggest, is field testing. They absolutely have to see how these extracts perform out in the real world. Under variable weather, rain, sun, wind with other microbes in the environment, maybe other pests. Do they still work? Are they stable? That's critical. A controlled chamber is one thing. An actual field is totally different.
Okay, so field trials are number one. What else? Assuming they work in the field.
Then comes the challenge of scaling up. We're talking about agricultural waste. If this takes off, you'd need potentially vast amounts of carob leaves and pomegranate husks.
Where would all that come from?
Exactly. You need to figure out the logistics. Collecting the raw waste material, processing it consistently to extract the active compounds, formulating it into a product farmers can actually use, distributing it, and it has to be cost-effective, right? Can you do all that at a price farmers can afford? Studying the industrial feasibility, the supply chains, that's a huge undertaking on its own.
So a lot of work still needed before this becomes mainstream.
Definitely. Promising, but still early days in terms of widespread application.
Okay, so what does this all mean for you listening right now? I think it signals a really interesting shift, doesn't it? Moving away from relying solely on synthetic chemicals and towards smarter, more biological solutions, often rooted in this idea of a circular economy. The key things to take away from our deep dive today. First, regulations, like those in the EU, are really pushing innovation in agriculture toward greener options. Second, scientists are finding ways to use natural compounds from waste to hit very specific weak points in plant diseases like those fungal spores and infection structures. It really does feel like agricultural waste could become a valuable resource, maybe even one of our best new tools against crop diseases.
I think that's spot on. And if we connect this to the bigger picture, it shows this convergence, right? You've got regulatory pressure meeting scientific breakthroughs. It's driving us towards farming systems that are not just about controlling pests, but about being smarter with resources, recycling co-products, and building more inherent resilience.
It's a whole system approach.
Exactly. And maybe here's a final thought to mull over. We saw that the carob extract worked partly by triggering the olive tree's own defenses, that antioxidant response, the phenolic compounds. So the really provocative question is, what other common agricultural waste materials, things we overlook every day, maybe grape pomace or almond hulls or citrus peels, what hidden potential do they have? Could they be sitting there waiting to activate completely different but equally crucial defense pathways in other important crops?
finding the next generation of crop protection not in a lab synthesis, but maybe hiding in plain sight in the waste stream.
It's a fascinating possibility. The potential for discovery in what we currently just throw away, it could be enormous.